Okay. So the next couple of slides, I need the lights turned down, so that's why we did that. Any questions on anything I've covered in the first hour or so? Okay. So next section is a section I think is pretty cool, and that is fluorescent in situ hybridization, otherwise known as FISH analysis. So I'll typically describe it as FISH as opposed to saying fluorescent in situ hybridization every single time. So this was developed probably about 20 years or so ago now, and it really revolutionized in many respects how cytogeneticists view chromosomes. We are actually now transitioning into a new era in terms of visualization. This has been kind of the gold standard, but it's now in the process of being replaced, unfortunately. But I think it's pretty cool in terms of how we look at chromosomes. So understanding this technique, I think, is important to understand what you are seeing when you're looking at these things. First of all, we have a chromosome spread on a glass slide. So there's nothing different about that process of isolating the cells, getting them onto the slide, getting a nice spread. Then we do two things with the chromosomes on that slide. The first thing we do is we just stain with a general DNA fluorescent stain that gives us a background appearance for the chromosomes. And so this blue kind of appearance to the chromosomes, that's our background stain. Otherwise, if you didn't do that, you couldn't see the chromosomes at all. So that's the background. And the background can be different colors. In this particular one, it's kind of a bluish color. Then we have a specific DNA probe. And that DNA probe is then labeled with a fluorescent molecule. So the DNA has a fluorescent tag. And that fluorescent tag is going to be a different color than our background color. And in this particular slide, the fluorescent color is this reddish color here. So we take these chromosomes that are on the slide, we denature the chromosomes so that they are single-stranded. So the chromosomes adhere to the glass, but they're single-stranded as a result of using probably an acid treatment. That opens them up. Then we take those and we add this fluorescently labeled molecule, this fluorescently labeled DNA molecule, and we allow it to hybridize to the chromosome spread. And it will hybridize wherever it matches. And we can do different stringencies in terms of how specific we want this hybridization to be. But in this particular case, our probe is a fluorescent probe for the lamin B receptor gene. And so then what we are seeing is here is where the lamin B receptor gene is localized on that particular chromosome. Now, good question is, why are there two dots? Why does that chromosome have two distinct dots for the lamin B receptor gene? Why isn't it just one? And the best answer for that is whenever you're looking at chromosomes, chromosome spreads, you're dealing with replicated chromosomes. They're in metaphase. They have gone through S phase. So every single chromosome consists of two sister chromatids. And so you should be able to detect both spots. Sometimes they merge together where you can't quite see two distinct spots. Sometimes you can. And then here is the second one. We have this one and then a very faint one there. And that is kind of hard to explain. That's probably an artifact. As to why that spot is showing up, I don't know. But that is the process. Once we have done that hybridization, we look at it under the microscope, and obviously we can detect this. So, the point. Very, very good technique for looking for subtle abnormalities in chromosomes, what are called microdeletions. And there are a number of what are called microdeletion syndromes that involve very small, relatively small changes in the DNA that we can detect with this particular technique. That with normal karyotyping, you probably would never be able to see because the deletions are too small or the duplications are too small. So we commonly use this now to look for gain or loss of genetic material. Here is another example. So what we are seeing here is we're using two different probes. And one of those probes is for the centromere of this chromosome. And so it's going to identify this chromosome, and that's centromere, and this chromosome and that centromere. And then the second probe is for a gene that's on that chromosome. And you'll see this chromosome is hybridizing for that particular gene, and this chromosome is not. That tells us that that chromosome, the lower chromosome, this one down here, has suffered a deletion, which has eliminated the gene that we are probing for that this one detected. So therefore, that person has lost that particular gene. This is usually way too small to see with G-banding or even high-resolution G-banding, because they're on the order of, they sound large, usually hundreds of thousands to millions of base pair, but it literally takes tens of millions of base pair to see it in actual karyotype. So this spread is for a disorder called Smith-Magenis syndrome. It's on chromosome 17, and it involves a deletion on chromosome 17. Good question based on this is Smith-Magenis an autosomal dominant or an autosomal recessive disease? Or syndrome, I should say. And how would you know? Autosomal recessive diseases or syndromes, you must be homozygous. For a dominant disease, heterozygous. So is this homozygous or heterozygous? It is heterozygous. Right? One chromosome has a deletion, the other one doesn't. The person has Smith-Magenis, therefore it's a dominant disease. So, two probes, chromosome 17 specific, and then a probe that we know is deleted in Smith-Magenis syndrome. This is the kind of the clinical presentation of Smith-Magenis syndrome, and this should be a hyperlink for you as well. And it takes you to a site that is called OMIM, and OMIM is Online Mendelian Inheritance in Man, and it is a really useful site. And I'd highly recommend at some point in time you go to OMIM, omim.org. It is essentially what it is. And it has a particular site where they say clinical phenotype. And that's what you really want to click on. It has a lot of genetic information that is probably too detailed for a medical student, but the clinical presentation is very, very useful. So you can see that there's a number of different systems that are affected in this kind of disorder, suggesting that probably there's a number of genes that are deleted in Smith-Magenis syndrome. This is another disorder called Williams syndrome. The gene that we are screening for here is for the elastin gene, but Williams syndrome has the elastin gene plus others that are deleted. It's larger than just the elastin gene, but we use that particular gene to detect it. And here we've done something a little bit different, and it's better, where we have two different fluorescent probes. One is green, and it is for the centromere, so it's highlighting that one, and then it is probably highlighting this one as well. And then there's red, and the red is for the elastin gene. And so you can see that this chromosome is hybridizing for the elastin in red. This chromosome is missing the hybridization. So again, we have a deletion of chromosomal material, and I forget which chromosome it is. I think it's chromosome 8. No, chromosome 7. So the elastin gene plus other genes had lost. And that then results in Williams syndrome. You could go to OMIM, put in Williams syndrome, and see then the clinical presentation associated with Williams syndrome. One of the characteristics of Williams syndrome, which is kind of interesting, and it's been in the news recently, I don't know if people saw this, but I'll mention it because it's kind of cool, especially because you had the dogs visiting yesterday, is Williams syndrome, people who have Williams syndrome tend to be super affectionate. And super affectionate for most everybody. You know, they don't have that kind of filter for strangers that most people do. And so they kind of represent what I wish we all were like, right? And this is the story that was recently. What makes dogs so friendly? Study finds genetic link to super outgoing people. The super outgoing people are those who have Williams syndrome. So they have identified deletions in one of the genes in dogs that is also in that same region in people who have Williams syndrome. So the idea then is that there was this artificial selection that humans imposed upon dogs So all dogs essentially have Williams syndrome. Not sure that that's true or not, but it's an interesting hypothesis. And because it made them so friendly, it was strongly artificially selected for by humans. So when you see a really friendly dog, they may have that. It's kind of cool. If you see a dog that is just a pain in the ass, then different. And probably humans have caused that too, most likely. So fish can also be really nice for detecting excess genetic material. Oh, one other point that I was going to make is if you are interested in this career of being a fish analysis type person and you are colorblind, forget it. It's not going to happen, unfortunately. I don't know if we have any individuals here. About 8% of the male population are colorblind. Is that red and green are the predominant colors that are there. And so just not going to be in the cards for you. So here we are looking at a gene that has been amplified in a cancer cell. And so the background stain is red. Propidium iodine is the background stain. And then this particular chromosome has had a huge amplification of that genetic material. And so we can see that. I think this is a c-Myc gene. And I don't know if you've heard of c-Myc before, but then that overexpression of c-Myc is what is driving that particular cancer. And I should have read my slides because it's right there. One of the things you'll note here, if you look at this carefully, you see hybridization in other regions as well. And you say, well, what is this other stuff? It looks like kind of nonspecific. One of these is the other chromosome, and it may be this one. But others are c-Myc is representative of a gene family, meaning that there are a number of different genes that have similar sequence to c-Myc. And so when we use this probe for c-Myc, it's picking up Myc-related genes. And so that's what some of these other hybridizations are identifying, is sequences similar to c-Myc. Chromosomal abnormalities. This is really nice for picking up chromosomal abnormalities. When we have an individual who has sex chromosomal abnormalities, for instance, too many X chromosomes, so here would be detection of 49XXXXX karyotype. Is that possible? Yes, it is. Is it rare? Very rare. But it is possible. One of your maybe take-home assignments will be, how could you get a person who is 49XXXXX? How would that work? And right now, I'd say you don't have enough information to be able to answer that question. But as we go through this, hopefully you will have enough information that you could piece that together. So we then use FISH for this. And what we have is a centromeric probe for the X chromosome. It is specific for the X. So then we have this spread, and we hybridize it with propidium iodine, that's our background stain, and then our X chromosome, centromere probe. And then we count 1, 2, 3, 4, 5 X chromosomes in this particular spread. This is how it used to be done with the bar body, and you may have heard of a bar body before, where you would actually have to do that. And this is a 47XX female, and there's 2 bar bodies, and it really challenges you to identify bar bodies in this. It takes a level of skill that is significant. Whereas here, the level of skill is not so great. 1, 2, 3, 4, 5. Pretty simple. You can also do this on interphase cells. This is really a nice feature as well. You don't have to wait for cells to be in M phase. You can isolate metaphase cells and do this hybridization. So this is a disorder that's called Charcot-Marie-Tooth disease. Has neurological symptoms plus other things, so especially a peripheral neuropathy. And what we are doing is we have a red probe for DNA that's typically deleted or duplicated in CMT. CMT is a duplication disorder. And then we have a green probe for chromosome 17 because the CMT region is on chromosome 17. And essentially, this is one of the 2 chromosomes. We can't really see it, the chromosome itself, but we can see the hybridization. So there's the centromere here, and there's a spot for the CMT. And then this chromosome has the centromere, and then it has 2 spots. These are interphase cells, so they're G1. They have not gone through DNA replication. So there aren't sister chromatids in these chromosomes. So one spot represents the one gene on that particular chromosome. 2 spots, duplication of the CMT region. And that would be a diagnosis of Charcot-Marie-Tooth disease. This is especially useful when we're looking at fetal tissue that's been isolated by amniocentesis, for example. Because one of the things that has to be done with amniocentesis, when we talked about preparing chromosomes, is you have to proliferate the cells. You have to grow the cells. And it takes time before you can get enough cells to be able to do the analysis. If you can just do it directly on the isolated amniocytes, you can do it much, much faster. And so this technique has really sped up the ability to make a diagnosis based on amniocentesis and based upon the cells that we've isolated. Here's another one. This is, again, interphase. And what we identified here is 47XXX, where, again, we have an X chromosome centromeric probe. We have 3 hybridizations in each of these interphase cells. And so, again, we've made the diagnosis of 47XXX, triple X syndrome, this one is called. The next thing, and we can turn the lights up at this point, is what is kind of taking the place of FISH. And it has a hard, difficult name, microarray-based comparative genomic hybridization, ACGH. And this is similar to FISH, but it's different in some respects as well. And now I can't see it. And it is really changing things also. This is kind of the future of cytogenetic analysis. Many labs are beginning to do this, although certainly not all of them. So this is similar to microarray. Have you talked about microarray before? I can't really tell. No? Some people have probably heard of microarray, but not enough to really go. So microarray. Microarray is where, on a glass slide, we have taken individual segments of DNA and we put them on little microdots. And we know exactly where each and every one of those dots is. So what we do in this particular case, here's our glass slide. Each one of these dots represents a different part of the chromosome. But the really cool thing is that it's a true linear array. So if we were to go along this entire length, we would go from the telomere of the P arm of chromosome 1 to the telomere of the Q arm of chromosome 1 in a linear fashion. So each of those dots represents, in succession, a part of chromosome 1. And then the next one is maybe chromosome 2. And then the next one is chromosome 3, chromosome 4, chromosome 5, etc., all the way down. So we have dots that then represent the entire karyotype, represented by maybe 15,000 dots on this slide. We take then patient DNA and we take control DNA and we fluorescently label it, just like we did with FISH analysis, except that we are fluorescently labeling all of their DNA. Okay? So we have the microarray, all of the chromosomal dots. We have the patient DNA that we think maybe has some sort of a chromosomal abnormality. We label that green. This one we label red, fluorescence. We mix the 2 samples of DNA together. So they are combined. And we are very careful in terms of making sure that the total amount of DNA between the patient and the control is equal, that they're balanced in terms of total DNA. We then take that mix sample and we apply it to our microarray slide. This DNA has been denatured, so it's single-stranded as well, open for hybridization. So the labeled probes here are going to then compete for hybridization to those spots. And they will try to bind. But if a red color is already bound, then a green one is not able to because it's now double-stranded. So there's competition. So, and that competition is really important. So let's say that there's a particular chromosomal region where between the patient and the sample, it's different. Let's say the person is trisomy 21, for example. So the patient has 3 copies of chromosome 21. The control has 2 copies of chromosome 21. So any probe that is fluorescently labeled from chromosome 21 is more abundant in the patient sample than the control sample. Which one is going to hybridize at a higher amount on the slide? It's going to be the one from the trisomy 21. So if there is a difference, the color of the hybridization is going to be different between these two. If it is equal, then there is an equal amount. If it's different in one way, DNA damage loss, in this particular case, it hybridizes more predominantly red. And if it is gain, it hybridizes more predominantly green. You certainly have to keep track of which one is which when you do these kind of studies. So a deletion or duplicated region is going to compete differently for hybridization to the glass slide. We then take these glass slides and we basically allow the computer to do its work. It analyzes each and every spot in terms of the intensity of that spot. And then it's going to give you a readout. And so here is, for instance, chromosome 7, and this is the plot of hybridization. And what we are seeing is that it's a good mix right at the center line. It's a good mix, and then we come to a point where there is divergence in terms of the hybridization color. One goes up, the other goes down in that particular region of the chromosome. And then it comes back again. So we can map this back. We know exactly which spot that is. And then we can map that back to the chromosome, and then we can say, okay, there is loss of genetic material in that particular region. A common question is, well, how do you know it's loss versus gain? Is you know which way it's going to shift based upon how you set up the analysis. And so we can do things like this. So here's the schematic of the chromosome. Here's the fluorescent pattern that we see. Usually we don't have a probe for the centromere, so it skips over that region, and then we see a shift in that chromosome. So that designates an abnormality in that particular region. Here's another one where we're seeing the hybridization. Everything is normal. We don't see hybridization here because it's the centromere. We're coming along, and then we have a clear shift in band 1p36.21. Duplication, deletion. In this particular case, it's a deletion. Here's another one where the normal fluorescence, and then we have a distinct shift in a very small region. And they actually went back and they did FISH analysis on this to confirm that, yes, that chromosome does in fact have a deletion, 17q, that takes out that region. And so by the time you guys get out there and practice, in terms of what is done, it's going to be a CGH, largely. I think this is going to just simply take over because it is such a very, very specific and very detailed technique, and even better than FISH analysis. So this is another analysis where, in this particular case, what we're seeing is early, very early onset schizophrenia in this family. Childhood onset schizophrenia. And we do a pedigree, and it appears to be what looks like a dominant mode of inheritance. And we do this kind of analysis. And what we see is that patient 617 on chromosome 3 has a shift in the P25 region. So that shift is there. We analyze 618, who is the other parent. They do not have that shift. Then we analyze the offspring. 621 did not have the disorder, no shift. 620, no shift. 619 had the disorder and has the shift. 622 doesn't, and they do not have the shift. 499, which was the proband, has the shift. So there's a very clear alignment or correlation between inheriting the chromosome that has that particular deletion or duplication, in this particular case, the duplication in that region and the development of early onset schizophrenia. You can go back into this region and say, okay, what are the genes in that region and how might they contribute to schizophrenia? So very useful, very valuable techniques, I think, that we're going to see increasing in utilization as time goes on. Any question about that technique before we go on? Yes?
How long does that technique take?
How long does it take? And how much does it cost? It's another good question. The technique, I would estimate probably about 10 days to 2 weeks, so not a long period of time. So not that much different than a typical lab analysis that is done. Right now it's probably pretty expensive, but as this gets automated, the price is going to come down. So this is most likely going to be the method of choice as time goes by. So next thing we want to talk about, specific chromosomal abnormalities. So we have a number of different terms here. First one is polyploidy. When you see the word ploidy, that designates an entire chromosome set. So polyploidy would be the gain of one or more entire sets of chromosomes, of haploid chromosomes. Monoploidy is the loss of an entire chromosome set. These are terms that are popular in terms of plant genetics, not so much with humans, because we really don't see those kind of things, at least in surviving humans. I'll show you some examples where it can be seen in at least conceptions. Cells can also contain chromosomal sets that are not a multiple of 23. And this is typically the gain or the loss of single chromosomes. So loss of the X chromosome, the gain of chromosome 21. These are oftentimes called aneuploidies, not ploidy, aploidy. And there's two essentially types, monosomy. And so when you see the term somy, you're dealing with usually individual chromosomes that are either gained or lost. So monosomy, one chromosome. Presence of a single copy of a particular chromosome. The rest of the chromosomes are all normal. Diploid. And then trisomy. 3 copies of a chromosome, with the rest all being normal in terms of 2 copies of the chromosomes. You can also sometimes see triploidy. And triploidy, ploidy here, chromosome set. This would be 3 sets of chromosomes within the nucleus. 3N, in other words, 69 chromosomes in total. Is this compatible with life? No, it is not. Is it possible to see it in conceptions? Yes, and very commonly. And then tetraploidy is very rare, but it is essentially 4 copies of the chromosomes. So you think of the nucleus as being jam-packed with a diploid. Think about it with 92 chromosomes. Monosomy. The only monosomy that you need to worry about in terms of actually seeing it in a patient is monosomy for the X. Very few, only a few cases of autosomal monosomies even surviving to term. And those that do survive to term do not live more than hours, if they survive at all. X, oftentimes it's designated with an O. That's kind of going away, where they just designate it as 45X. So 45X monosomy is Turner syndrome. And this is seen in about 1 in 2,500 to 1 in 5,000 live female births. So it's a pretty frequent chromosomal abnormality to see. Monoploidy, never seen. Trisomy, we'll talk about. Triploidy is seen in about 1 in 10,000 live births, and so we'll talk about that one. And then tetraploidy does happen in conceptions, but it's extremely rare for a conception to survive to term because it has a very, very high rate of lethality during development. So polyploidy at conception. Triploidy, 1 in 10,000. If you were to do a karyotype, this is what you would see, where each and every chromosome is present, rather than 2 copies of that chromosome, 3 copies of that chromosome. The vast majority of these are spontaneously lost. I think I lost a word there somewhere. Are spontaneously aborted. Most common cause of this is what's called dispermy. Dispermy is the simultaneous fertilization of an egg by 2 sperm. I don't know if you've gone through embryogenesis at all in terms of this, but typically once the sperm fertilizes the egg changes in terms of its membrane to prevent a second sperm from being able to fertilize that. But if 2 sperm come in basically at the same time, that it is possible that the two sperm could contribute their genetic material to that single egg. And so dispermy would cause then a triploid conception. The vast majority of these, when they occurred, do not survive to term. But they are a very common cause of early pregnancy loss. So 69XXY would be the karyotype that you would see here. And I moved the X and the Y here just because I didn't have enough space and I wanted to fit it in. So, aneuploidy. Aneuploidy, where one of the chromosomes is abnormal. Trisomy 21 is an aneuploidy, for example. Most common cause for this is what's called non-disjunction. And non-disjunction is definitely a process you want to be aware of in terms of what happens. So, this occurs during meiosis. And having a basic understanding of meiosis is really helpful here. We won't talk a whole lot about meiosis. I'm assuming that you have at least a basic understanding of meiosis. Is that there's two stages to meiosis. So we take this cell right over here. This is looking at just one homologous pair. In a human going through meiosis, we have 23 chromosome pairs. We're looking at just one of them. So what should happen in meiosis 1 is the homologs separate from each other. And that's what has happened normally here, is the two homologs have separated. One homolog to the left, the other homolog to the right. So one possibility is non-disjunction during meiosis 1. And essentially what that means is that this cell received both of the homologs, and this cell received neither of the homologs. Now, one danger with showing just one chromosome is that this shows a cell that has no chromosomes, and that is absolutely not the case. This cell over here has every other chromosome because it separated properly. The only one that didn't separate properly was this one, and so that's the one we're showing. So it's important to realize this cell has every chromosome except this one. This cell has all of those chromosomes plus an extra copy of that one. So it has these two homologs. Then these cells will go through meiosis 2. And in meiosis 2, the sister chromatids separate from each other. And so the assumption here is that this process occurs normally. So this cell will go through meiosis 2. The sister chromatids will all separate. This one will go through meiosis 2. The sister chromatids will all separate. So one of these two will go to this cell. One of these two reddish chromosomes will go here. And then for the blue ones, one of these two sister chromosomes will go here, one will go here. So what you will see here then is that this particular gamete, either sperm or egg, has an additional extra chromosome. If that's chromosome 21, this cell now has 2 copies of chromosome 21. Then we have fertilization. So we'll assume that this is happening in a female. And this would then be a germ or an egg. That egg will be fertilized by a normal gamete from the father. It could be the other way around as well. That sperm is going to bring in 1 copy of that chromosome, assuming everything went properly for the male. And normally it does. This is a rare event, so the likelihood of it happening twice is kind of infrequent, but possible. This will bring in that second one. So if this particular sperm fertilizes this egg, it will have 2 copies of every chromosome except for this one. And it will be trisomy for this chromosome. So this would then be trisomy. If this sperm fertilizes this egg, then that particular conception is going to be monosomy for that particular chromosome and disomy for all of the other ones. So in offspring, or conceptions is maybe a better term for it. In conceptions, you can see trisomy conceptions and monosomy conceptions if you have non-disjunction in meiosis 1. Now, the other slide of this. Here, we have these 2 chromosomes disjoined normally in meiosis 1, but then there was non-disjunction in meiosis 2. In meiosis 2, the sister chromatids are supposed to separate. Both sister chromatids went to this cell. Neither went to this cell. If this one gets fertilized, monosomy. If this one gets fertilized, trisomy. And then the question is that compatible with life? The vast majority of trisomies are not. And there's only selected ones that are. And we'll talk about the ones that can survive. But this is the process of non-disjunction, failure of the chromosomes to disjoin normally during meiosis. Meiosis 1 versus meiosis 2. One other kind of take-home project for you on this one is there, can you tell the difference? Would you be able to tell the difference between meiosis 1 non-disjunction and meiosis 2 non-disjunction? Notice that they both produce trisomies and monosomies, but is there something different about a non-disjunction in meiosis 1 versus a non-disjunction in meiosis 2? And hopefully you can see that there's a difference, but I'll leave that for you. This non-disjunction can occur in either sex. So it can occur during male meiosis or female meiosis. So the most common one we talk about is trisomy 21. So 47XY plus 21 or 47XX plus 21, obviously depending on the sex. About 1 in 800 live births is trisomy 21. This is one of the most common at birth in terms of chromosomal abnormalities that you can see. They have a characteristic phenotype. That characteristic phenotype is described as Down syndrome. So genotype is trisomy 21. The phenotype is Down syndrome. And distinct facial characteristics are one of the first clues with respect to the diagnosis or the suspicion of a trisomy 21. And then they would do a karyotype analysis to verify that that is true. Individuals who have Down syndrome and have a number of physical difficulties, mental retardation, to varying degrees. There's certainly varying levels of functionality with respect to individuals who are trisomy 21. They oftentimes have GI tract obstructions, especially early on. Congenital heart defects are very common and can be life-threatening at birth. Ventricular septal defects are especially prevalent amongst individuals who have Down syndrome. A much higher risk for leukemias later in life, and AML and ALL, acute lymphocytic leukemia and acute myelogenous leukemia. Don't have to know that now. But those are the two leukemias that are more common with Down syndrome individuals. They can also develop Alzheimer's-like symptoms. You ever heard of the APP gene, amyloid precursor protein gene? APP is localized to chromosome 21. And so the Alzheimer's-like symptoms are thought to be due to overexpression of the APP gene from chromosome 21. Males are invariably sterile as well. And so one of the things that, if you recall, that I wanted you to be able to do is you should certainly be able to recognize a Down syndrome phenotype based upon a description of that individual. One of the things that they oftentimes have, and it's designated here too, is if you look at your own hand, you typically have two predominant creases that go across your palm. And an individual with Down syndrome tends to have just a single crease in their hand as opposed to two predominant ones. If you see a child with Down syndrome, you'll oftentimes see that they have very thick glasses as well. And so eye abnormalities are quite common also. And hearing loss is predominant. 10% of all mental retardation in the United States is due to Down syndrome. And so this is quite significant in terms of our population. Origin of the extra chromosome. 95% of cases of trisomy 21, the extra chromosome is contributed by the mother. And this is a very important point to keep in mind. Advanced maternal age. Advanced age of the mother, starting at approximately 35 years of age is where it increases. This is the most significant risk factor for producing a child with trisomy 21, is advanced maternal age. Probably have heard that before at various times. This is showing the incidence of trisomy 21. So this on the y-axis is frequency of Down syndrome and maternal age. And you can see it's very, very low until we reach about age 35, and then it begins to increase. Another thing you'll note is that there's a demarcation here at amniocentesis in red at birth in blue, and there's a difference between those two. One of the things that is true about trisomies, and this is true for trisomy 21 as well, is that they have a significantly higher rate of spontaneous pregnancy loss. So a female who is carrying a trisomy 21 conception is at higher risk of losing that conception than a conception that does not have a trisomy. That's true for monosomy as well. So even though trisomy 21 is compatible with live birth and survival, there's a much higher rate of pregnancy loss that is seen. 20 to 25% of trisomy 21 conceptions survive to term. So a very large percentage of them do not. And again, karyotype, 3 copies of chromosome 21. You should definitely be able to pick that out from a karyotype. Boards like to show you various karyotypes and say, what's the most likely abnormality in this patient, or something like that. Trisomy 18. Trisomy 18, the phenotype for trisomy 18 is called Edward syndrome. It is the most common chromosomal abnormality amongst stillborns. So amongst conceptions, trisomy 18 is more common than trisomy 21. It is more lethal during embryogenesis. So the incidence among live births is only about 1 in 6,000. So the vast majority of trisomy 18 conceptions are spontaneously lost during pregnancy. Characteristics for those that do survive. Low weight for gestational age, diaphragmatic hernia, significant mental retardation. They tend to have fingers that overlap, and you can see this even at birth where they present with overlapping fingers. I'll show you some images of this. Clenched fist, small eyes and a small mouth. Heart defects, a small receding jaw, short sternum, malformed ears, tend to be low-set ears as well. Developmental retardation, and this is unusual, short big toe that bends upwards. It's a very common characteristic also. Kidney defects and omphalocele. What is omphalocele? I feel like it's Friday instead of Tuesday. No, what day is it? Today's Tuesday. Omphalocele is where the essentially central GI, and I'm not an anatomist, I'm sure an anatomist could give a much better designation to this, develops outside the body. And so it's kind of like a hernia, but it isn't a hernia because it formed that way from the very beginning. 50% of Edwards syndrome children who are born will die in the first month. 90% will die within 1 year. So this is a very poor prognosis for a child when they are born. Characteristics that you can see the overlapping fingers in these individuals, characteristic facial presentation, malformed ears, clenched fingers, or a clenched hand with overlapping fingers as well, and then karyotype chromosome 18, 3 copies of chromosome 18. 95%, similar to trisomy 21, 95% non-disjunction in the mother. Is it correlated with advanced maternal age? Yes. So the same thing holds for Edwards syndrome as it did for Down syndrome. Trisomy 13. The phenotype for trisomy 13 is called Patau syndrome, about 1 in 10,000 live births. I'll give you the characteristics. You should be familiar with these characteristics. You can read these the same as I can read these, so I'm not going to read them off to you. But mental retardation and developmental retardation are 2 of the major common characteristics seen. Patau syndrome, rates of survival very similar. Very high rate of spontaneous loss of the conception for trisomy 13. And again, maternal contribution to the extra chromosome, 90 to 95%. Maternal age playing a role as well. And then this one gives a nice presentation. Cleft lip is one of the things that's kind of unique in terms of Patau syndrome. It's commonly seen as well. Postaxial polydactyly, commonly seen. You see a clenched hand just like you saw before. But here, if we count the digits, 1, 2, 3, 4, 5, and then an accessory digit. And then here as well, 1, 2, 3, 4, 5, 6, an extra digit. Postaxially, so postaxially polydactyly. And then karyotype is going to show 3 copies of chromosome 13 for Patau syndrome. So, maternal age and autosomal trisomy, Down syndrome, Patau syndrome, Edwards syndrome, they all exhibit a higher risk for advanced maternal age. Lots of different things have been looked at in terms of what correlates. And maternal age is the one that correlates absolutely the best. It's the strongest predictor of this. So for women less than 30 years of age, the risk of trisomy is about 1 in 1,000. At about 35 years of age, it's up to about 1 in 350 conceptions. For women age 40, it's about 1 in 70. And then for women age 45, it's about 1 in 25. So it goes up and it goes up significantly as time goes by. So, good question. Are there more Down syndrome births in older women above 35 or younger women, 35 or below? Where do we see most of the Down syndrome births in the population? And older would be the most obvious incorrect answer, unfortunately, is that it's younger. Why? And you have to look at risk. Certainly the risk of producing a Down syndrome child is much higher in a 45-year-old woman than a 25-year-old woman. 1 in 25 versus 1 in 1,000, approximately. Why in the younger population? Numbers. A lot more 25-year-old females having offspring, having children, than 45 years of age. What this figure shows is two things. One is the relative risk of trisomy with maternal age, and then this line represents the number of births that occur in the population. And so we can see that the number of births peaks at around 25 years or so of age, maternal age, and then declines as time goes by. One of the things that we are most definitely seeing is a trend toward later women, older, having offspring. So as the number of females who have offspring at 35, 40, and perhaps beyond increases, this is something that definitely has to be kept in mind for these individuals, is that they are at increased risk for having these 3 different trisomies. This is why one of the things of the idea of banking eggs at an earlier age is a consideration. Because if you bank the egg when you are 25, as long as it is preserved properly, when you then thaw that egg out, that's an egg from a 25-year-old instead of a 40 or 45-year-old. So that's a possibility as well. So about 75% of all children who are born with Down syndrome are born from women under the age of 35. So when you talk about genetic screening tomorrow, we'll talk about methods for trying to identify those trisomy conceptions in women who are younger than age 35, because that's where the majority of these kind of conceptions are occurring. So, more than 90% of all children are born to women under the age of 35. Sex chromosome abnormalities, the last thing I'll talk about. We won't quite finish. Actually, we have about 50 seconds left. I'm going to stop here. So we'll pause here. We'll take a break for 10 minutes. When we come back, we'll talk about sex chromosomal abnormality.